All right, so welcome to the last session for today. So this talk is going to be given by Dr. Trilochan Bhagarthi. So Trilochan did his PhD in statistical physics from the Institute of Physics, Bhubaneswar, and then followed it up with a postdoctoral stint at Harishchandra Research Institute, and then his second postdoc at this institute. So during this time, he did a lot of very fascinating biological modeling work, some on epidemiology, some on pattern formation. In particular, what he's going to talk about today is on slime mold. And so we are going to have like an overdose of slime mold tomorrow when Professor R. Bhaskar from IIT Madras and Professor Vidyanand Nanjundya will both be speaking on slime mold. So this is like your first glance at the system, which you'll have a lot of in-depth view tomorrow. So Trilochan is right now, Trilochan then subsequently moved to industry, and right now he's with Tata Steel at Jamshedpur. And we really wanted him to be present here in person, but because of some work commitment, he couldn't make it. But he nevertheless managed to join online. And so it's with great pleasure that I invite Dr. Trilochan Bhagarthi to give this talk. So Trilochan, over to you.
Thank you, Sitabra, and good afternoon from Jamshedpur. So this box is visible. Should I minimize this? So, okay. So I'll be talking about.
Trilochan, you might want to make it full screen. The
full screen. Yeah. Okay.
Yep. Good.
Okay. So I'll be talking about the collective dynamics and pattern formation in slime mold. This was my first work with Sitabra at IMSC many years ago. And recently we revisited this problem and we found some very interesting thing that can still be done here. So actually slime mold is everywhere. There are around 100 species, and everywhere you look, you will find slime mold. They are very fascinating single cell organisms, and they are mainly some species which are very fascinating. I mean, they form very fascinating patterns due to their collective behavior. So I'll be talking about mainly the modeling part that we did in IMSC. So this species is called Diptyostelium discorium, and this is the life cycle of this dipty amoeba, and they there are some spores that is generated from this bulb, and initially when there is sufficient food, they live as single cell organisms, but as food is scarce, they start starving, and when they start starving, they start forming this kind of aggregation colonies, and then this aggregation grow further, and here they form a slug, and from this slug there is a fruiting body, and again this stalk kind of structure forms, where again these spores are generated when this bulb is exploded. So this is the life cycle of the amoeba, and here I will be talking only about this part of the life cycle, where how single cell amoeba interact among themselves and form this dendritic structure. So our problem is, how do they interact and form this aggregation? So this talk I will be covering the introduction to this model and some of the ingredients of this model, which is the Fujinagumo oscillator and Langevin dynamics, and then I will discuss how this pattern are formed. So as a simplified mechanism, this cartoon will explain what actually happens when amoeba start starving. When these amoeba start starving, they secrete a cyclic AMP molecule, which is a SOS molecule, and this molecule can diffuse in the on the surface. And when this molecule is detected by another amoeba, that amoeba becomes active. And then when they become active, they again start secreting this CM molecule, and this CM molecule keeps on getting secreted more and more. And depending on the concentration of this CM molecule, the movement of this amoeba are decided. So how do they secrete this CM molecule is given by the internal dynamics of the amoeba, which is a relaxation oscillator. And in our case, this is the Fujinagumo oscillator. And with every spikes, there are some formation of CM molecule, which is secreted from the surface of this amoeba. This CM molecule then diffuses and then moves to other uh, inactive amoeba, and that is how it goes on. So earlier, there were some model, and here I will discuss a model by Vasiev. And in this model, what they have done is they have a continuous variable for the CM molecules. So the CM molecules, they diffuse in space, but uh, the internal dynamics is implicitly included in this diffusion equation. So you can see that there is no oscillator that is oscillating. Of course, this part, the reaction part here in this equation, if you translate it, it will turn out to be a Fujinagumo oscillator only, which actually Vasiev has mentioned in his paper. And then there is a diffusion of this amoeba, which is given by this equation, and a drift term that arises due to the recovery variable R, and a gradient of the CM molecule. So, so the amoeba and both the CM molecule are modeled here using continuum equation, and this is the pattern that is formed when you simulate this set of equations, and you will see that there is a very nice dendritic structure which forms a blob here at the center. Now, after many years, Nurberg et al. Has tried to model it explicitly using the relaxation oscillators. And you can see that this set of equations, AI and RI here, that is actually a Fujinagumo oscillator for each of the amoeba cell. So every amoeba here is acting as a relaxation oscillator, and the stimulus to this oscillator is described by the concentration of the CM molecule, and the CM molecule, the concentration uh, is governed by this equation. But you can see here that there is no diffusion part. So in this model, the amoeba are static, and there is a overall CM concentration which has some governing equation, and there is a source term which appears due to the activation variable. So what actually is happening here is, let's say we have a amoeba here, and every time it spikes, this term will kick, and there will be production of CM. And then when this CM concentration increases beyond a certain threshold, then we know that there will be oscillatory solution, and then those of the amoebas will start spiking. So this is the basic structure of Nuremberg's model. Here, the only problem is that the amoeba has no motion, and the overall concentration of CM is governed by this equation, which has no diffusion part. So in our model, what we want to incorporate is the discrete nature of the cells and their motion. Sorry, uh, the diffusion of CM molecule, and how we will couple the diffusion of CM molecule to the discrete motion of these uh, amoebas. So these two questions are the uh, main problems in our model. So it's one can say it's a mixture of Vasiev and Nuremberg's model. So what we do is, let's assume that we have n amoeba at position x1, x2, x3, xn. Each amoeba is a Fujinagumo oscillator, so therefore we can write down the uh, Fujinagumo equation for each of the amoeba, and the stimulus is given by the concentration of Cm in the neighborhood of the ith amoeba, and the motion of the amoeba is governed by overdamped Langevin equation, which is this form, where the force term is given by motility function here, which is same as that in Vasiev, and it depends on the gradient of the Cm concentration. And then Cm concentration, we have a diffusion term, a diffusion equation, which is a diffusion term and a source term. And then source term depends on the activation variable AI, and it is a point source. So you can think that there are n points in 2D space. Each point can generate some Cm molecule with a density rho, and this Cm molecule will diffuse, and whenever this Cm molecule is detected by amoeba in its neighborhood due to this IS function, it will start spiking. And then when it starts spiking, depending on what the force is at near this amoeba, you have a Langevin equation that governs its motion. So this is the basic structure of our model. So before I go to the result of this model, I just want to explain how this Fujinagumo oscillator works. So, so these are the null lines of the Fujinagumo oscillator. We can equate these two equations to 0 and plot the A null line and the R null line. And we see that this cubic, due to this cubic equation, there are three regions. And if we are in this region, which is less than this minima point of this cubic equation, any trajectory, anywhere we start, it will go and converge to this equilibrium point. So this means that there will be no spikes for this region. Similarly, if we take uh, this point, we will see that here also we will have no spike. This will again converge here, but here we will have a very flat kind of behavior of AI and RI. That is, a continuous function. There will be no oscillation here. So in between these two lines, if this linear curve intersect anywhere in between those two lines, then we can have very pronounced oscillation, and that will lead to the spiking. Second, we also need to see how the Langevin dynamics describe the motion of the amoeba. So let's say we have a overdamped Langevin equation like this, with eta the noise term and f is the force term, or the systematic part of the noise. And the noise, the mean of the noise is 0, so there is no biasness in the noise. And the correlation is given by some constant times delta function. So this d here is the diffusion constant of the amoeba. As you can see here, we have dA. So if we discretize this, we can write it as this form, and then we can take mean, and we, as we know, the mean of the noise is 0. We will have a very nice form, that is dx by dt is mean of so mean of dx by dt of mean of x is mean of fx. Similarly, we can square both sides, and then we can take the mean, and we can see that xt times eta will go to 0, the mean. But the square of mean, from here we can see that if t goes to t prime, we can write this delta t minus t prime as 1 over square 1 over delta t, and then this term will become 2d by delta t. And then we can write down the equation for the mean and the variance as these equations. So only this term will survive, which is of the order delta t. We will take delta t square order term to 0, and this term is there. So we have this equation. From this equation, one thing we can say is that the variance will grow linearly with time, because here we have a constant term. So some functions times something will be there. So this is acting like a source here. So if we simulate this Langevin equation, what we will see is that there will be some kind of this random trajectory, and as we go forward in time, the width of this trajectory, uh, the the extreme points, that will be square root of dt. So this t is inside the square root, and then at every point on this trajectory, if we discretize, we can write this in this very simple form, that is xt plus dt equal to xt plus the first term plus square root of this 2 dt times gamma t. Now here gamma t is a unit normal random variable. So how this happens, you can figure out very easily. That is, if you add two normal random variable, and whose variance is of the order 2 dt, then the variance of both of them will be squared. So this is the main the property of a normal random variable, and this can be found in D.T. Gillespie's book, or there is a very nice American Journal of Physics article where you can find this. So this is the Langema dynamics implementation. So this is how we have implemented it. Now, how to solve this model numerically? So here we have one equation uh, that is a continuum equation where we have a del square operator here, and here we have two ordinary differential equations. So there are three n ordinary differential equations, and n out of which n are Langevin equations. So this equation needs to be solved in R2, and this we can solve it in on a grid where there needs to be some approximation of how these terms will be coupled. So let me describe it here in this grid. So now suppose these are the grid points. I will draw some box around these grid points. Then I can solve the diffusion of the CM molecule on these grid points. But I have a source term which depends on the position of the amoeba, and the position of the amoeba can be anywhere because it's in the continuum. So what I will do, I will draw a box of length delta x, and any point inside this box I can evaluate this source term, and depending on the state of the oscillator, this theta will be either 0 or 1. So this is how this is done. But there is also a force term. So suppose I have to calculate the force term for this amoeba. But then to calculate that force term, I have to evaluate this gradient of the CM molecule. And it also depends on the recovery variable. So which I can, since the amoeba, since it's the amoeba where it's sitting, so I don't have to worry about this term. But the gradient term can be again calculated on this grid. So what we do is, let's say I have an amoeba here. So I will draw a box. Let's say this, for this amoeba we can discuss. So what will happen is, we have 4 points, and any amoeba inside this box, we have to calculate the gradients at these 4 points using, there will be 1, 2, 3, 4 in the x direction and 4 in the y direction. So that the gradient on this amoeba is the mean of all the gradients at these 4 points. So I have to calculate at each of these 4 points using, I think, 12 points. So there will be 4 plus 4 and 4 plus 4. So sorry, 8 points. No, actually it will be 12 points. So that is how this force term can be calculated. And then we can integrate the Langevin equation. And then I think now I'll show some patterns which we have recently simulated. So here we will see some very nice dendritic structure, and which depends on the diffusion constant of the CM, and the diffusion constant of the amoeba, and the mobility factor. So the maximum mobility factor comes from this VR function. So that is also parameterized. So I have taken three of these parameters. So that is diffusion constant of CM, diffusion constant of amoeba, and the maximum mobility factor. So here, what I have done is I have assumed that the CM molecule diffuses very fast, and of the order of one order higher than the amoeba. So if we run this simulation, you will see that very nice dendritic structure starts evolving, and this spiral pattern is very coherent, and the amoeba particles moves toward the higher CM concentration. So as time progresses, what you will see is a lot of this amoeba will be aggregated at this center. So this is this nice structure that forms. So next, I'll show another. Oh. So here, what I have done is I have increased the motility of the amoeba. So for the same gradient, now the amoeba can move much faster, uh, 10 times, sorry, twice as fast as the earlier, and we'll see what will happen. So first thing that we can observe here is that they form this kind of blobs first, and these blobs start moving. Actually, if you increase the motility factor, even much larger blobs can be formed. And then this nice dendritic structures will start evolving. But now you see that at some point, the spiral structure will break, and it is no more a coherent spiral that is there. And this is how it will evolve. So as time progresses, what will happen in this structure? What we can see is that there will be lot of small colonies, which is contrary to what we earlier found. Earlier we had a one single colony at the center, and now these amoebas are rushing towards multiple colonies because the spiral structure has broken. So I think this is all. Um, thank you for your attention. I am afraid it was very short. Uh, Maybe I'll take questions.
Okay, let's thank Trilochan. Questions?
I can't hear you.
Trilochan, can you hear us? Maybe.
Yes, yeah, yeah.
Okay, okay. Yeah, so this, we are asking for questions. So, Saling.
Hi, Trilochan.
Hi.
Um,
Yeah.
so the pattern that you get, for amoeba density. Is that a steady state pattern?
So this is in the steady state. For the first video that you saw, what will happen is they will gather together at the center and they will form a single colony. All the dendrites will be gone. So that is what will happen.
You end up with a blob, single blob finally. Is that that a steady state pattern?
Sorry?
So will the steady state pattern be just a single blob?
Yes, yes. Yeah.
I see.
So something that we found very recently, and I was discussing with Sati, and maybe we need more investigation. That is, if you assume that this DA is the slide visible?
Yeah,
yeah,
it's visible.
So this DA here, if you assume that this is constant, then in the steady state, you will not have a blob, a single blob. But if you assume that this DA depends on the density of the amoeba, that is, if you take some sigmoidal function where for low density, the amoeba diffuses faster and many order of magnitude less when the density is beyond certain threshold density, then what you will see is a single blob at the center. Otherwise, this blob will form, but after a certain time, you will see that they will spread out and they will form a constant kind of structure. They will not form very nice blobs. That is what numerically we have found.
I see. And if this chemotaxis happens in a fluid medium, then you would expect the patterns to be much more rich, I suppose.
Yes.
Right now, it's a solid substrate, right?
Yeah. So in this these two simulations, the density depends on the diffusion coefficient depends on the density.
Right. Thanks.
Any other questions? Shakti.
Yeah, this is just more of a comment. I mean, I'm guessing since a lot of people here might not be that familiar with excitable media, maybe you could just talk about how you sort of set up the spiral wave in the first place. Just maybe explain.
Yeah. So so this is should I start from the model or the Fujinagmo oscillator?
Yeah, sure.
Yeah. So initially, what we do is we take a uniform distribution of the amoeba on the grid. I mean, on the surface, and each one of them has some random A and R variables. So they have some values, and then depending on what the A's and R variables are, if you see from this null lines, if you increase the stimulus I, then this cubic will shift up, and as it shifts up, this point will shift right and it will come to the second region. And whenever that happens, you can have sustained oscillations of all these spikes. So this function depends on this CM concentration, has a threshold. If it increases beyond certain threshold, I will increase sufficiently, and then that amoeba will spike. Once it spikes, this source term will pick up. It will act as a point source. It will start giving out all this CM molecule, which will diffuse in space. And depending on what the density field is, this force is decided for each of the amoeba here, and then they start moving according to what the force is at that point, and that is how this structure is formed. So I hope that is correct. I mean, is that okay?
Yeah, yeah, that's fine. Thanks.
Any other questions or comments? Okay, if not, let's thank Trilochan once more.
Thank you.
Thanks,
I missed.
Trilochan.
I could have come. I like more of a blackboard talk, but yesterday I was talking to Sakti and I was very scared because I was thinking that there will be less slides. So Sakti told me to go slow and talk more. But I'm some other time I'll cover the main details of the
Sure,
model. Yeah.
sure. Anyway, tomorrow, like there will be two talks, one by Professor Bhaskar and another by Professor Vitan and
Yeah.
Nanjun on slime mold. So it's good that, you went first so that, they'll now see how that model fits in with reality.
Yeah.
Okay. Thank you so much,
Thank
Trilochan.
you. Thank
Okay,
you for the attention and hope to meet you all sometime.
sure.
Thank you.
Then, yeah. Okay, bye.
Bye. Bye.